Every time you take a bite of a fresh slice of pizza or, I don't know, lean in to smell a flower on a morning walk, you probably think you're just having this passive experience.
Right, like it just happens to you.
Exactly. You take a breath, you chew, and sensation just sort of, it just is. But in reality, your face is acting as this massive, instantaneous chemical data processing lab. So welcome to the deep dive.
Thanks. I'm really excited for this one.
Yeah, today we are going to completely dismantle how you think about your senses of taste and smell. We're going to figure out exactly how the body detects flavor and odor and separate some deeply entrenched historical myths from what the biology actually tells us.
It really is a complete paradigm shift once you understand the actual mechanics of it. I mean, to start, we have to establish what we actually mean when we say a chemical scent.
Right, because it's not like seeing or hearing.
No, not at all. If you think about vision or hearing, your body is fundamentally detecting waves.
Yeah.
Light waves bouncing off objects into your eyes, or sound waves vibrating through the air into your ears.
Oh, okay, yeah.
But taste, which we call gustation, and smell, olfaction, they are entirely different beasts. They are built to detect actual physical molecules in your environment. The scientific term for this is chemoreception.
So it's not enough to just like be in the same room as the pizza. Actual physical contact between the food and your cells is required.
Yeah, exactly. For you to taste something, the molecules of that food have to be physically dissolved in your saliva.
Oh, wow.
Right, that liquid medium is what allows the molecules to wash over and physically touch the specialized receptor cells on your tongue. And then, for you to smell something, volatile molecules, meaning, they're light enough to have evaporated into the air, they have to be sniffed or wafted up high into your nasal cavity.
Dip them into the nose.
Yeah, and once they're up there, they physically bind with a patch of specialized tissue called the olfactory epithelium.
Okay, let's unpack this. So if the mouth and nose are basically a high-tech chemical testing lab for the body, how exactly does the machinery work? I kind of want to start with the mouth because the mechanics of taste seem a bit more contained, like a good baseline before we get into the wild complexity of the nose.
I think that's a great place to start. Taste gives us the foundational logic of how a physical molecule floating in the world triggers an electrical nerve signal to the brain.
Right.
So the physical architecture starts with the papillae. If you look at your tongue in a mirror, those are the visible little bumps.
The ones everyone thinks are the taste buds.
Exactly. A lot of people mistake those bumps for the taste buds themselves, but they're actually more like housing units.
Yeah.
The taste buds are tucked inside those bumps, and inside those taste buds are the actual microscopic taste receptor cells.
And from what I understand from the sources, these receptors aren't just a one-size-fits-all situation, right? They use completely different chemical mechanisms depending on what they're trying to detect.
Right. So let's look at sweet, umami, and bitter. These are detected by complex proteins that sit on the surface of the cell. They're G protein-coupled receptors. Think of them like a highly specific lock on a door.
Okay, lock and key.
Yeah, when a specific molecule, the key, binds to the outside of the cell, it activates the lock. This sets off a chain reaction inside the cell, specifically using a protein called gustatory, which eventually fires a signal to the brain. It's this intricate, multi-step communication relay.
But there's something about this architecture that really stood out to me. When you look at the genetic breakdown of these specific receptors, there is a huge, glaring imbalance.
So the bitter receptors.
Yes. The sources say we have roughly 25 different receptor types specifically tuned to detect bitter compounds, but we only have one primary receptor pair for sweet. If human beings are so incredibly driven to seek out sugar and sweet things, why do we have 25 times more hardware dedicated to bitterness?
It's all about evolutionary survival. I mean, you have to think about this from the perspective of early humans foraging in the wild. Sweetness signals energy.
Right, calories.
Exactly. It tells the brain, hey, here are carbohydrates, here are sugars. That chemical profile in nature is relatively narrow. Sugar is sugar. But bitterness is nature's universal warning sign for poison.
Ah, okay.
And there are far, far more ways for a plant to be poisonous than to be nutritious.
Right, because plants can't just get up and run away. They have to fight back with chemistry.
Precisely. Over millions of years, plants have evolved this massive variety of complex, toxic compounds to stop animals from eating them. Because those poisons take so many different molecular shapes, our bodies needed to evolve a much broader, diverse array of biological alarms to detect all those different potential threats.
So we need a wide net for poison, but just like one reliable sensor for energy.
Yeah, exactly.
That makes total sense. But what about salty and sour? Because you mentioned those don't use that complex lock and key relay system.
Correct. Salty and sour operate much more simply, using what are called ion channels. Instead of a multi-step relay, think of an ion channel as just a physical gate in the cell wall.
Just a literal open door.
Pretty much. For a salty taste, when you eat a potato chip, you literally have tiny Gates ENSC channels on your tongue that just pop open and let the sodium flow directly into the cell, triggering the nerve.
Wow.
And sour is similar. It's detecting the direct flow of acid, specifically protons. And what's amazing is that the exact acid sensor for sour was a scientific mystery for a long time. It wasn't until 2018, out of Emily Lyman's lab, that researchers finally identified the precise channel, the OTT1 channel responsible for it.
Wait, really? It's incredible that we were still identifying the basic mechanics of sour taste in 2018.
I know, it sounds crazy.
Which actually makes the story of umami feel a lot less unusual. The timeline of how we understand taste seems to have a lot of historical lag.
It really does.
Right.
I mean, the story of umami is a perfect example of that.
Right, because it was discovered way back in 1908. A Japanese chemist named Kikunae Ikeda was eating his wife's kombu dashi broth, which is made from kelp, and he realized there was this rich, savory taste that just didn't fit into sweet, sour, salty, or bitter.
Yeah, he knew it was something distinct.
Right, so he went into his lab, isolated the specific molecule responsible, which was glutamate, and he named the taste umami. He even patented a way to produce it commercially. But Western science essentially ignored or debated his discovery for almost an entire century.
Yes, it took until the early 2000s for the scientific community to fully validate it. It wasn't until researchers, specifically Zucker and Reiba, finally isolated the exact genetic receptor pair for umami that the debate ended.
They found the actual hardware.
Exactly. They proved at the DNA level that we have dedicated hardware for this savory taste. Ikeda was right all along. And, this raises an important question. If the five-taste textbook definition can change after a century, is it still changing right now?
It absolutely is. The boundaries of what counts as a basic taste are actively expanding.
Exactly. For instance, in 2015, researcher Richard Matz proposed a potential sixth basic taste called oleogustus.
Oleogustus, that sounds intense.
Right. But this is not the texture of fat, not that creamy mouthfeel of ice cream, but the actual chemical taste of fat molecules themselves. They are actively studying a specific receptor, CD36, just for that. There's also preliminary work looking into whether we have dedicated sensors for water and even for the carbonation in a sparkling drink.
Okay, so the tongue gives us these basic fundamental survival categories: energy, salt, acid, poison, and savory proteins. But that only paints in broad strokes. Because, if you blindfold me and give me a lemon and then give me a lime, my tongue's sour receptors are firing the exact same way. It's just acid.
Yep, the hardware sees the same thing.
But I instantly know the difference, which means we have to move beyond the broad strokes of the tongue and look up into the nasal cavity.
We do, because to understand the infinite variations of the world, taste just isn't enough. We have to understand smell.
Here's where it gets really interesting. For a long time, nobody actually knew how the nose distinguished thousands of different odor molecules. We just didn't seem to have the genetic toolkit to categorize every single smell in the universe. But that paradigm broke open in 1991.
Yes, thanks to a landmark discovery by Linda Buck and Richard Axel, which eventually won them the Nobel Prize. Before them, olfaction was just a black box, but they discovered a huge family of genes that encode our olfactory receptors.
So they're somewhat like those lock and key proteins we talked about with taste.
Similar, yeah, but they are expressed high up in the nose.
How does the actual wiring work, though? Like, how do the signals get sorted so my brain knows what I'm smelling?
So the overarching rule of the nose is one neuron, one receptor. Humans carry roughly 400 functional olfactory receptor genes. So any individual sensory neuron in your nose will express only one of those 400 types.
Okay, just one.
Right. Now, here is the incredible part of the architecture. Every single neuron in your nose that expresses the exact same receptor type sends its physical wiring, its axon, to the exact same microscopic destination on the olfactory bulb in your brain. These sorting stations are called glomeruli.
Okay, wait. Let me make sure I'm picturing this correctly. So a chemical signal is turned into a spatial map in the brain. Is it kind of like a piano, where you only have a set number of keys, in this case 400 receptor types, but by striking different combinations of those keys all at once, you can play a nearly infinite number of complex chords?
What's fascinating here is that your analogy is exactly how combinatorial coding works. If one receptor only detected one specific odor, We could only ever smell 400 things.
Which isn't a lot.
No, not at all. But biology is much smarter than that. One receptor type can respond to multiple different odors, and one single odor molecule can activate multiple different receptors simultaneously.
So it's the pattern.
Exactly. It's that unique combination of the specific chord being played that allows those 400 genes to encode vast amounts of information.
Which naturally leads to the big debate in the scientific literature, right? Exactly how many unique chords can we hear? Or, well, how many smells can we detect?
Right. Back in 2014, there was a very widely publicized science paper by Bushted and colleagues that used this combinatorial logic to estimate that humans can discriminate over 1 trillion different olfactory stimuli.
1 trillion. I mean, that sounds mathematically impressive, but biologically impossible.
And many scientists argued exactly that. Shortly after, a rigorous rebuttal was published by Gherkin and Castro. The critics argued that the statistical method the original team used was completely flawed.
How so?
Well, they had basically taken a very small sample of odor tests and extrapolated it out exponentially. The critics demonstrated that if you manipulate the math on a sample size that small, you could produce almost any massive number you wanted, depending on your assumptions.
Oh, wow.
Yeah, it's a really great lesson in how a genuinely amazing biological principle, combinatorial coding, can sometimes get compressed into a headline-grabbing number that completely outruns what the biological data actually supports.
Okay, so we have the tongue giving us the basic survival categories, and the nose giving us this incredibly complex combinatorial map of the world. But when you and I sit down to eat a meal, we don't feel like we're experiencing two separate senses running in parallel. It just feels like one unified experience.
And that unification is arguably one of the greatest illusions your brain performs every single day.
Really? An illusion.
Oh, absolutely.
Yeah.
When you experience the complex taste of a meal, you are overwhelmingly experiencing what is called retronasal olfaction.
Retronasal, so smelling from behind.
Exactly. Let's break down the mechanics. When you chew your food, you are breaking it down and releasing volatile odor molecules. When you exhale, those molecules don't just disappear. They travel up the back of your throat, through the nasopharynx, and into your nasal cavity from behind.
So we're basically smelling our food from the inside.
Yeah. Your brain takes that complex scent profile, merges it with the basic sweet or salty data coming from your tongue, and projects the entire combined sensation onto your mouth as flavor.
Which perfectly explains why food tastes like absolutely nothing when you have a severe cold. Your tongue is still perfectly capable of detecting sugar or salt, but that retronasal channel in the back of your throat is blocked by congestion.
Yep, the air can't carry the odors to the nose.
So you lose the food's identity entirely. The brain can't build the illusion without both data streams.
Exactly.
Now, speaking of that illusion, I've always heard people throw around the statistic that flavor is 80% smell. But looking into the history of that claim from our sources, it turns out it's basically a media-generated pseudo-statistic. There is actually no peer-reviewed origin for that exact 80% number.
It's very true. Now, flavor scientists absolutely agree that retronasal smell dominates our perception of flavor. It provides all the nuance. But you can't just slap a clean 80% measurement on an experience that's subjective.
Right, it's just not that simple.
No, flavor is a highly complex multisensory construction.
Which brings up a sensation that has always confused me. What about the physical burn of a chili pepper or the icy coolness of a strong peppermint? If it's not a basic taste on the tongue, and it's obviously not a smell in the nose, what sensory system is actually processing that?
That is an entirely separate sensory system called chemoreceptors, and it operates through the trigeminal nerve.
Yeah.
What you are experiencing are chemical-triggered touch and pain sensations.
Wait, touch and pain, so spice isn't a flavor at all.
Not technically, no. Let's look at the mechanics. The active compound in a chili pepper is capsaicin. Capsaicin physically binds to a specific receptor, the TRPV1 heat channel on your nerve endings, that is literally designed to detect dangerous physical heat. Like if you sip boiling water.
Oh, man.
Yeah, the chemical tricks the nerve into firing a heat warning. Conversely, the menthol in peppermint binds to the TRPM8 channel, which is designed to detect physical cold.
So it's basically a false alarm.
Completely. These pain and temperature nerves ride right alongside your taste and smell, signals to the brain, creating the ultimate multisensory experience. But when you eat salsa and your brow starts sweating, your body is literally reacting to a phantom heat burn, not a taste.
So the brain is just being hacked by a chemical. Honestly, because flavor is such a convincing, multifaceted illusion, It makes sense that our scientific history is completely littered with assumptions that became entrenched as false facts.
Oh, there are so many of them.
I read that even Aristotle got confused back in 350 BCE, lumping taste and smell together conceptually because they are so deeply intertwined when we eat.
And that confusion set a lasting precedent. Because our chemical senses are so subjective, happening entirely inside our own heads, they are highly susceptible to myths that go unchecked for decades.
Which brings us to the biggest myth of all, the one almost everyone listening probably learned in elementary school science class, the tongue map.
Oh, the famous tongue map.
Right, this is the idea that you only taste sweet at the tip of your tongue, bitter at the very back, and sour on the sides. Where did that map actually come from?
It has a very traceable and very bizarre origin. It comes from a 1942 psychology textbook by a researcher named Edwin Boring. Boring was reviewing data from a 1901 German dissertation by a man named David Hannig.
Okay, so far so good.
Well, Hannig had simply measured extremely minor differences in how sensitive different parts of the tongue were to certain tastes. We're talking about tiny, negligible shifts in detection thresholds. But Boring misinterpreted the translated data. He drew a diagram showing these areas as exclusive, fixed zones.
So it was just a terrible game of academic telephone.
Precisely. And what's wild is that in 1974, a researcher named Virginia Collings directly retested the premise and completely debunked it.
1974.
Yeah. She proved experimentally that every single region of your tongue that has taste buds can detect all of the basic tastes. The regional differences in sensitivity are so tiny, they are practically meaningless. Yet because the map was so visually simple to teach, it stayed in textbooks for decades.
That's wild. Another massive historical misuse of science we have to talk about is the public panic around MSG.
Ah, yes, the so-called Chinese restaurant syndrome.
Right. This whole thing started in 1968 when a physician named Robert Ho Man Kwok wrote a letter to a medical journal. He described experiencing temporary numbness, palpitations, and weakness after eating at Chinese restaurants, and he pointed the finger at monosodium glutamate, or MSG.
And the historical record is very clear on how wildly this spiraled. That single anecdotal letter sparked decades of public anxiety and literally shaped food labeling worldwide.
But the science didn't actually back it up.
No, the rigorous science tells a fundamentally different story. First, MSG is simply the sodium salt of glutamate. Glutamate is a naturally occurring amino acid that is highly abundant in tomatoes, Parmesan cheese, mushrooms, and even human breast milk.
And looking at our sources, when researchers actually ran controlled, double-blinded studies, they largely failed to substantiate or reproduce a consistent physical reaction to MSG at normal dietary doses in the general population.
Exactly.
The literature also notes, and we're just reporting what the sources highlight here impartially, that food historians have heavily scrutinized the racialized undertones of this panic. The anxiety was directed almost entirely at Chinese cuisine, while completely ignoring Western processed foods that were loaded with the exact same glutamates. It's really a classic case of a subjective anecdote becoming public fact much faster than the scientific method could catch up.
It is. And honestly, it's not the only time we've underestimated our own biology based on bad assumptions. Take the long-standing scientific belief that human olfaction, our fundamental sense of smell, is incredibly poor compared to other animals.
Oh, right. The idea that because we evolved to be highly visual upright primates, we somehow just lost our chemical edge.
Exactly. That myth traces all the way back to a 19th century comparative anatomist named Paul Broca. Broca looked at the human brain and noted that our olfactory bulb is proportionally very small compared to the massive size of the rest of our brain, especially when you compare it to, like, a dog or a rat.
Okay, so it looks smaller relative to the rest of the brain.
Right. Because of that visual proportion, he argued this made us non-smellers, with a biologically vestigial sense of smell.
But modern anatomical research completely refutes that.
Completely. In 2017, neuroscientist John McGann published a landmark paper proving that absolute bulb size does not dictate olfactory ability. When you actually count the cells, humans have just as many olfactory receptor neurons as many other mammals.
So we have the hardware?
We do. And when you run controlled behavioral tests, humans can match or even beat other species on specific scent discrimination and tracking tasks.
We're actually incredibly good at it. And speaking of biological differences, we don't even all taste things the same way as the person sitting next to us. There is fascinating work by researchers like Linda Bartoshik on genetic supertasters.
Yes, the genetics of taste are highly individualized. There is a specific gene, the TAS2R38 gene, that codes for a bitter receptor. and variations in this gene dictate exactly how intensely you experience certain bitter compounds.
So some people really are tasting a different world.
Oh, absolutely. People classified as supertasters physically possess more of those housing bumps, the fungiform papillae on their tongues, meaning a denser concentration of receptors. So they experience bitterness much more intensely, which fundamentally alters their preferences for things like broccoli, coffee, or alcohol. It proves that the flavor world you live in is uniquely your own.
So what does this all mean? We've talked about the architecture, the mechanisms, the debunked myths. It's easy to treat all of this as really fun cocktail party trivia. But when this intricate machinery breaks down, the real-world consequences go way beyond just missing out on a good meal.
If we connect this to the bigger picture, these chemical senses are actually vital clinical indicators. During the COVID-19 pandemic, anosmia, the sudden loss of smell, became a household word. It was one of the very earliest, most reliable signs of infection.
And researchers out of Robert Data's lab at Harvard finally figured out exactly why the virus caused that specific symptom.
Yes, they did.
Yeah.
And the mechanism is fascinating. They discovered that the virus wasn't actually infecting the delicate sensory neurons themselves. Instead, it was attacking the support cells, the sustentacular cells in the nasal cavity, which express the ACE2 receptor.
Oh, so it goes after the support crew.
Exactly. These support cells maintain the environment for the neurons. When the virus infects those support cells, it triggers massive local inflammation. Think of it like the scaffolding around a building suddenly expanding and collapsing. It physically pinches off and disrupts the neurons so they can't fire their signals to the brain.
But that's actually good news in the long run, right?
Right, because the sensory neurons themselves weren't directly destroyed. Once the inflammation went down and the scaffolding was repaired, most people eventually recovered their sense of smell.
But smell loss isn't just about acute viruses. It's also an incredible early warning system for long-term neurodegenerative diseases.
That's right. A reduced ability to smell hyposmia is now heavily recognized as one of the earliest prodromal signs of Parkinson's disease. It can appear years, sometimes a decade, before any of the classic motor symptoms like tremors show up.
A whole decade.
Yeah, this aligns with a major theory called Heiko-Brake's staging hypothesis, which suggests that Parkinson's pathology actually originates in the olfactory bulb and the gut, slowly working its way upward before it ever reaches the parts of the brain controlling movement.
It's a profound diagnostic window, not to mention the sheer everyday safety risks. As we naturally age, our chemical senses decline. Practically speaking, if you can't smell, you lose your biological early warning system for smoke in the house, a gas leak in the kitchen, or spoiled food in the fridge.
It is a critical life-saving function.
Yeah.
But beyond just physical safety, smell is deeply inherently tied to our humanity and our memories.
Ah, the famous Proust phenomenon, like smelling a specific brand of sunscreen and being instantly transported back to a childhood vacation.
Exactly. Smell has a unique anatomical privilege in the architecture of the human brain. Every other sense, vision, hearing, touch, has to pass through the thalamus first. The thalamus acts like a central switchboard, filtering and routing signals.
But smell skips the line.
It totally skips the line. Smell is the only sense whose primary signal bypasses that switchboard entirely. It projects directly into the limbic system, specifically targeting the amygdala, which processes emotion, and the hippocampus, which forms new memories. It is a direct, completely unfiltered wire to the emotional core of your brain.
Which perfectly sets up the final thought I want to leave you with today. We've spent this time seeing how eating and smelling are not passive events, but incredibly complex, life-saving chemical data processing systems. But tucked away in the margins of the biology is a profound paradox.
It really is one of the most beautiful mysteries of the nervous system.
Consider this. Unlike the neurons in your eyes or your ears, your olfactory receptor neurons are completely unique because they are constantly regenerating throughout your entire adult life.
Yeah, they live for a few weeks, die off, and are replaced by fresh cells.
Which means the physical biological cells you are using to smell your morning coffee today are not the exact same cells you used to smell it a few months ago. So if the very hardware of your nose is constantly dying and being reborn, how do the emotional scent memories from your childhood remain so perfectly, permanently intact?
It's a breathtaking question. It shows just how much we still have to learn about the brain.
It's definitely something to ponder the next time you take a bite of your favorite meal or catch a familiar, nostalgic scent on the breeze and realize just how much unseen, microscopic machinery is working tirelessly to build that experience for you. Thanks for joining us on this deep dive. We'll catch you next time.